On behalf of our department and then the School of Science and Technology, I welcome you all to this seventh lecture of this academic year. I would like also to thank Agilent Technologies that have been supporting this lecture series since the start in 2007. Before I start, let me use the guest speaker for next time. It is Dr. Issam Maroof from Department of Electrical Engineering in San Jose State University, and his talk is titled Reaching for the Saturn System with Cassini Spacecraft. It's going to be an interesting one. For today, we have invited Dr. Rahul Singh from the Department of Computer Science of San Francisco State University, and his talk is titled Drug Discovery Against Neglected Tropical Diseases, a Computer Science Perspective. Dr. Rahul Singh is currently an associate professor in the Department of Computer Science at San Francisco State University. His technical interests are in computational drug discovery, bioinformatics, and multimedia information modeling and management. The primary thrust of Dr. Singh's research activities lies in developing algorithmic techniques and computational infrastructure that are especially of value in curing the neglected diseases of humankind. Dr. Singh received his PhD in computer science from University of Minnesota and a diploma in computer science from the Moscow Power Engineering Institute. Prior to joining academia, he was a principal staff scientist in the area of imaging at Cymagics Incorporated. Earlier, he founded and headed the computational drug discovery department at Exelixix Incorporated. At Cymagics, he was the co-designer of the Protein Mind software, which received the Frost and Sullivan Technology Innovation Award. He was a San Francisco State University Presidential Fellow 2006 and is a recipient of the Carrier Award from 2007 to 2012 in the of the National Science Foundation. So here is Dr. Rahul Singh.
Thank you. Gentlemen, if my vision doesn't mistake me, and the lady, thank you for inviting me. It is truly a pleasure to be here. And I think you mean with this. All right, so I apologize. I was down with a cold, which has been going around the Bay Area, and I have recovered, but my voice has not completely recovered yet. So normally I don't sound this awful, but hopefully we'll manage somehow this 1 hour. So when I was deciding upon the title of this talk, I wasn't sure. It sounds like a very eclectic sort of title. And once I came here today and Ali was describing the background of the students here, I thought that maybe I should have said an engineer's perspective, and that would have been equally accurate. Okay. So what I want to actually talk about a little bit today, for those of you who are especially students and actually all of us here, are some very revolutionary ways in which science is progressing, in the sense that the disciplines and the boundaries within which we study things when we are as students, as you will see more and more, they cease to exist as you go and become professionals in the area. So actually, if you look at the future, it is quite possible that science education and scientific research is going to be organized in manners very different than how, for instance, I was taught when I was a student. And hopefully, through this talk of mine, you will get some insights into how the barriers between classical disciplines are breaking down and new sciences are occurring really at the interface of disciplines. Okay? So, I assume that most of you here, and correct me if I'm wrong, but you're mostly not familiar with the notion of neglected tropical diseases. Is that correct? All right. So I'm not speaking to an audience which are experts, who are experts in the area. Good. So let me actually try to motivate what we are really facing here and why this is an interesting problem. So, I'm sure you've all heard of diseases like HIV, tuberculosis, malaria, right? And if you think about it, these are the so-called big three diseases of our planet. Okay, so for instance, tuberculosis inflicts, probably greater than 2 billion people. That means 1 in every 3 person on this planet is inflicted by this disease. HIV/AIDS is much smaller, it inflicts about 35 million people, still a very large number. Malaria, which has now become a focus of a lot of research activity, inflicts up to 500 million people. So these are the big three diseases. What I want to talk about today is, what people call the neglected tropical diseases and what I like to call the great forgotten diseases of mankind. Okay? So these are diseases, of these types. There's a disease called leishmaniasis, trypanosomiasis, or sleeping sickness, Chagas disease, and the particular disease I'm going to focus on today is called schistosomiasis. So if you look at these diseases, schistosomiasis inflicts over 200 million people, and over 500 million people are in danger of being afflicted by this disease. Okay? And if you add up all of these diseases, you get to a very, very big number. So why haven't we, as not specialists, but just as educated people, why haven't we typically heard about these diseases? And the reason is that in spite of the fact that so many people suffer from them, these are primarily diseases of the poor people. Okay, so essentially, these are diseases of people who do not have credit cards. Okay? And because of that, it is just not very feasible in the business models of pharmaceutical companies to spend billions and billions of dollars to try and create drugs to cure these diseases because you can't make a good profit out of it. And that's fully understandable, but the fact remains that we have hundreds of millions of our fellow human beings on this planet inflicted by diseases which we don't tend to study too much. Okay. So I think from purely a moral perspective, as educated people, as people who have the opportunity and the potential ability to make real changes to civilization, this is not a state of affair I think any one of us would be comfortable with. Right. So let's go a little bit more and look at the distribution of these diseases. So in particular, I'm looking at schistosomiasis, and I'll talk a little bit more about this disease, what causes it, and why it's such a fascinating disease. But one of the things you will see is that this is a disease which is endemic in Africa, in South Asia, in China, parts of Southeast Asia, in South America, and as a matter of fact, the parasite exists even in California. But interestingly, it does not recognize the human as a host. So if that change were to come, this disease could happen in California too. So what is this disease? So the term schistosomiasis actually means split worm. And I'll talk more about it, but basically, this is a disease where this parasite, and this is what this parasite looks like. This parasite has different stages in its life cycle, but it breathes inside snails, which are in the water. Okay? And if you're in this infected water, the parasites, when they mature, they get out of the snail, and then they sense for the human skin. Okay? And typically when they find the human skin, I don't know how well you can see this, this is a photograph of a part of a person's arm where this parasite entered the human body. And what it does is, it actually looks for the hair follicles, and it enters the body through the hair follicle. So it enters through the skin, it gets into the bloodstream, okay, goes to the heart of the lung, and then migrates to the portal blood vessel in the liver, and it matures there. Okay, so this is actually, I don't know how clear it is, but this is actually the image of a parasite inside the portal vein. Okay. So one obvious thing, of course, is that when we have a parasite and when this is mature, this could be as big as a centimeter or a centimeter and a half. So this is a big organism, right? And it's sitting inside your portal vein, so it's obviously going to occlude your portal vein, right? And you can have blood pressure because of that, you can have a stroke because of these occlusions. But what is very interesting is that this parasite is an enormously advanced creature. So I'm a computer scientist, but when I was shown a dissection of this parasite, I was amazed at the richness of his nervous system. A really well-defined nervous system goes all into the body. And what is very interesting is this parasite, once it gets into the body, once it mates for life. So these guys are monogamous. And this actually, what you see here, this bigger part is actually the male, and the smaller part is the female, which sort of lives for her whole life and just grooves in the male's body. Okay? And once they reach sexual maturity, they produce eggs. And they literally produce over their lifespan tons of eggs. As a matter of fact, the female, after sexual maturity, can produce an egg every 5 minutes. Okay? And how long do they live? This parasite has a life cycle of between 40 to 60 years. So this lives nearly as long as a human being. Okay? And they found this parasite inside the mummies of the old Egyptian pharaohs. So this is an old parasite, which has been around for a long time. Okay? And the problem is that this parasite, it causes some serious complications to the body. So one of the things is it causes this fibrotic granulomatous. It causes the tissue to change, and that can lead to a variety of different problems. It can cause enlargement of the spleen, it can cause bladder cancer, and it has also been correlated with lack of impeding the intellectual development of the affected people, especially children. Okay, so indirectly the parasite does not kill the host, the human, but it's what's called a debilitating disease. It will destroy your ability to be productive. So it has a huge economic impact in the areas it hits. So very interesting organism. So when you think about these guys, sometimes you wonder if we are really the top species on the planet. Incidentally, one very interesting thing I want to remind you is, think about the following. Let's say you have heard of people having transplants in their body, rejecting the transplanted organ or something, right? So the body has an immune system which rejects foreign things. These are centimeter and a half. They live in the body for decades, and the body immune system cannot reject them. They have found a way to fool the body's immune system. This is no joke. These guys are seriously advanced. Incidentally, one of the things people also think is once we figure out how to do it, we could maybe use the same ideas to ensure that the human body does not reject a transplant and so forth. Right? So there are a lot of different ways when we skin the cat about how we can get advantages out of our knowledge. But anyway, so going back to this disease, and I'm going to slowly transition into where engineering comes in, but I'm going to talk a little bit of chemistry and biology for a few slides. So there is actually one drug which works against this disease. Now this drug has been around for 30 years. It's called Praziquantel. It's a very good drug. You can give a single oral dose, has few side effects, has bioactivity against all 3 species of the parasite which cause the disease, and basically the drug is quite affordable. The problem is that if you have a big disease which is spread all over the planet, has millions of people afflicted, and you have only one drug against it, sooner or later you would start seeing drug resistance. And that is exactly not only the fear now, but literally, in the time I have started working on this disease, which is about two and a half years, we are now seeing evidence of drug resistance. So now in Egypt, for instance, we see evidence of drug resistance against the parasite. So, in addition to the fact that there is drug resistance, the drug does not cure all the parasites. It's more, potent against the adult parasite and less potent against the juvenile parasites, and so forth. So there are a lot of reasons why we need to develop new therapies against this disease. Okay, so now, if that sort of makes sense, let me sort of actually give you a one-slide introduction to how drug discovery is done today. Okay? So drug discovery is one of the most expensive industrial endeavors we take on these days. In the US, for instance, the average cost of getting a successful drug into the market is about $1.5 billion. And it takes about 15 years to get a successful drug to the market. And the failure rates are greater than 99.5%, depending on how you calculate it. Okay, so highly expensive, very slow, and high failure rates. Right? So if you think about it in terms of actual cost of, to bring the product to the market, the costs are very similar to what you would do if you develop a new microprocessor, or if you develop a new passenger airliner. Right? But the success rates there are 100% easy. Right? So it's a similar cost, but very low success rate. And this is sort of very interesting to, should be interesting to all of us, because whatever happens, I think all of us will at some point in our life take a drug. Okay, so this really matters to us. I may or may not use Facebook, just to pick a name. Okay, but I will at one point in my life take a drug. Okay? So anyway, so the way drug discovery is done today is what happens in these companies is you would start with a collection of molecules. And this is typically a very large collection. You would have millions of molecules. And then you would identify a particular protein or an enzyme which each of these small molecules will go and interact with. It's like your hand fitting a glove. Okay? And you try to find out, doing actual chemical experiments, which of these molecules goes and interacts with this protein or this enzyme. Okay? And because there are millions of them, this is done in miniature, in parallel, in these sort of plates, and it's called high throughput scaling. And once you find out which drugs goes and interacts with your target, you go and take those molecules, typically a few hundred of them, and try to chemically optimize them so that they have the right characteristics. And then, if you succeed there, you go into clinical trials, and then you have $1 billion drug. So I think two questions as scientists we should be all interested in. One is, well, if you're talking about millions of molecules, where do you get millions of molecules from? Right? I mean, I don't know about you guys now, but when I was doing my high school chemistry, which is where I stopped my formal education in chemistry, when we had to synthesize something, it was test tubes. Right? It took a lot of time to synthesize a particular compound. Right? So synthesizing millions of compounds would take thousands of chemists working hundreds of years. Right? So the way it is done is using a technique called combinatorial chemistry. So the idea is follows. You take a particular molecule which has some attachment points. So let's say that you have this molecule which has these 3 attachment points. And let's say you have 100 fragments, pieces of molecules, which you can attach at any of these 3 points. Right? So combinatorially, you can have 1 million different molecules. So any of the 100 here, any of the 100 here, any of the 100 here. And the reason why people design molecules like this, I would like to illustrate with this example. So this is from the time when I was in the industry doing drug discovery. So if you look at these 2 molecules, they should look very similar to you. Okay, if they don't, then you are an incredibly good medicinal chemist. Okay. They should look similar to you, they look similar to most medicinal chemists. So these 2 molecules were both hits against something called the CCR1 receptor, which is implicated in rheumatoid arthritis. However, if you look at this, this guy has a single extra methyl group, a single CH3 in it, extra. Okay? And this guy goes and interacts with the CCR1 receptor at concentrations of 44 nanomolar, while this guy interacts at the concentration of 5 micromolars. So we do not need to worry about what this concentration means, but simply the point that 44 nanomolars is an order of magnitude less than a micromolar concentration, and lower the concentration, better the drug. Okay, so actually this is a potential drug. This is junk. Okay, so the point is that very small distinctions in the structure of a molecule can cause very large distinctions in how it may or may not interact with a particular target of interest. Which is why, not knowing what would be the actual structure of the drug, people try to sample the space as finely as possible and try and test everything. Okay. Now, for schistosomiasis, the problem is we have no information on this stuff. We have no idea what molecule in the parasite we need to target. So what people have tried to do is they have tried to directly test the drug against the parasite. Okay, so you put the parasite in a test tube, put the drug in there, and see what happens to the parasite. Observe it for some certain amount of time, for days. Okay? Now, of course, if you do something like this, it's obviously going to be very slow. And because it's going to be very slow, and because it will require a lot of manual expertise and time and endeavor, it cannot be high throughput. okay? Which means we cannot look at millions of molecules, which means, again, the chances of our finding a good drug against this disease, a good new drug against this disease, is going to be much smaller. Right? So the engineering problem I'm going to talk about today is how we can take this idea of introducing the drug not against a particular molecule, but against the entire parasite in a high throughput setting by using engineering science, or computer science in my particular case, but engineering science. Really. Okay? So we want to automate and create a method which will allow us to test millions of molecules against these parasites without any human intervention and capture all the effects depicted by the molecule on the parasite. And the effects, they can be many. It can change the shape of the parasite, it can change the speed of motion of the parasite, it can change the texture of the parasite, and so on and so forth. So we want to be able to measure all of these changes automatically. We want to be able to reason about these numbers and find out what the drug is doing to the parasite. So that's really what we want. All right, so what is the twist in the problem? The twist in the problem is very interesting. So interesting, as a matter of fact, that when we found this out, people who have been studying this, so this is actually a video, you can see this, that's moving on. People who were studying this disease for decades, even they were quite surprised. So the idea is the following. So here, what you see is a group of parasites, all of them in the same environment, which have been exposed to this drug called Praziquantel. This is this gold standard drug. Right? And as you can see, in ellipses of different colors, you can see quite different effects of this drug on the parasites. Right? So the ones in red, these actually look nearly unaffected. I can tell you that. Okay? These guys have this bulge in the middle. These guys have sort of really become dark and they don't move too much. Okay? And they're sort of getting broken down. Right? So one interesting thing is that different parasites in the same environment, exposed to the same amount of time, react differently, show different phenotypes to the same drug. Okay? The converse is also true. Different parasites show different effects as a result of different drugs. So here is the parasite in its normal state, which is what some of those guys were looking like. This is the impact of a drug called K777. This is the impact of Praziquantel. So different drugs can elicit different phenotypes. The same drug can also elicit different phenotypes depending on the variability of the parasite. So it is very interesting. These guys are individuals. Right? And it's sort of understandable, right? I mean, if you look at humans, I mean, all of us may take one particular drug, and the drug will act differently on each one of us. And the same thing here. Right? But this is very important because if we have the case where the drug is not acting very well against certain certain elements in a certain parasites, then we should know this because these parasites will become resistant to this drug. So we need to factor these things in. Okay, so as I said, our goal is to facilitate some sort of engineering solution which will allow us to do high throughput screening of these parasites. Right? So I'm not going to go into some of the biology of how this is done, but suffice it to say it's a very complex process to prepare the parasites. This is some of the work which has been done by our collaborators at UCSF, and there's a lot of interesting sort of robotics they've prepared. So this was a sorter which will go and sort these parasites and put them into these little wells. So there's a lot of interesting stuff here which I just don't have time today, so I'll focus more on the engineering part of things. So what we want to do is we want to videotape the parasites. Okay? Then we would like to segment each parasite out from the video recording. We would like to track the parasites during the entire course of observation, be it minutes or hours or days. As we track the parasite, we would like to continuously monitor and make quantitative measurements of the parasite, of the size, of its color, how it moves, and so on and so forth. Okay? And then we get into a bunch of algorithmic issues where we will see, if time permits, how these quantitative measurements can be algorithmically analyzed. Okay? And then what sort of inferences we can draw from them. So let's start with the first problem, which is the segmentation of the parasites. Now, if you guys have looked at signal processing or image processing, one of the things you would have probably realized is that the cognitive visual system is incredibly advanced. Right? So when we look at this, we can say, oh yeah, these are the parasites. That's quite obvious to it. But to make the computer do this is a very tough problem. Sadly, when I started my PhD, it was a very tough problem, and it still remains a very tough problem. Okay? Makes you feel that you've not done anything in your life. But you know, these are great challenges. So one of the reasons this is very tough is because, first of all, you cannot assume a standard shape of the parasite. Right? Sometimes in biology, you'll see the people will say, well, cells are roundish, or neurons have a particular shape. Right? You can't assume this. As you can see right here, there's a lot of variability. Right? And different drugs will make the parasite look very, very different. The movement of this parasite is based on elongation and contraction of its musculature. So it doesn't move like a snake, it moves like this, okay, which causes, again, its shape to change. Okay? So you have different shapes in the same image based on how the different parasites and what stage of motion they are in. Right? Then you can also see in these sort of cases some inner anatomical structure, the gut of the parasite. And that makes this extremely hard because you start getting discontinuities in the image gradient there, which are not corresponding to the outlines, the edges of the parasite. Then you have this really tricky problem that these guys, for some reason, which I fully don't understand, but they tend to stick together. And I've asked these from people who study these parasites, I said, why do they tend to stick together a lot? And no one exactly knows why. Okay, but they tend to occur in clumps, and as an engineer, that makes my life hellish. I would love them to be separate, because I want to separate them, but this is tough. They all stick together. I do not know where one parasite ends and the other one begins. Okay? And so on and so forth. So, the first thing you do, I try with my students is to take standard methods and see how they work out on this. And they don't tend to work very well. Okay? So you miss edges by doing certain types of techniques called morphological segmentations. If you've done image processing, you may have heard of the watershed transform. The watershed transform does not work very well in many of these cases. These clumps stay together. Okay? Standard thresholding performs poorly, it puts lots of gaps inside these parasites. More advanced watershed techniques have also not shown to work. They don't tend to work very well. So we had to come up and design a new technique. So one of the things we did was this idea of what is called the region-based distribution function. So this is work done basically at Yelena Kovacevic's lab at CMU. So they have a different problem they're looking at, they have a different method. So we adapted the first part of the method, and what it does is it takes the pixel values and passes it to this sort of a function. And this is a sort of sigmoid curve, and it basically, the result allows us to basically figure out what is the foreground and what is the background in the image. Okay. So instead of sort of talking more about the math, let me just show you how this works. So this is a video of how quickly this method will find out where the foreground is, okay, where the parasite is and where the backgrounds are. So you saw it started with a random initialization. Every pixel in the image was initialized to a mask, and it very quickly converges onto individual parasites. And then it takes a few more, few iterations, and then you'll see it slowly will converge, and we would have the foregrounds separated from the backgrounds. However, we do not have all the parasites separated. So you see there are 3 parasites here, but this is one big chunk. Right? So we still have work to do. But what we know very quickly is what is the foreground and what is the background. So then what we had done is we had changed the threshold in that region-based distribution function to the point where we have a massive increase in the number of the foreground objects, and then use that and then done post-processing to separate the parasites out. And this was a paper we had published last year. But what happens with this method is that if you have noisier images, then this method gives you very cluttered results, which are very hard to post-process. So one of the things I'll briefly show you this today is a new method where what we do is we, instead of figuring out the particular threshold by analyzing this curve, we use the threshold as the value which optimally separates the background and the foreground distributions, assuming that they are normals. So it's a bimodality assumption. And when we do that, we get a much better separation of the foreground and the background. And then there are some other steps we do. So after the separation is done, we apply what's an edge detection operation to find out where the edges of the parasites are. And there is a lot of research in computer vision and image analysis and signal processing on edge detection. And one of the best or very well-known algorithms actually is an algorithm developed by John Canny in Berkeley. This was done actually when he was still doing his master's degree at MIT. But anyway, it's a very well developed, very well-known algorithm. Works in general very well, but here is an example where this algorithm doesn't work very well because of this inner anatomical structures of this parasite. So the edges of the parasites sort of have a thickness to it, which causes this edge detector to fail. Okay, so what we did was we sort of developed a new method where the key steps of the Canny edge detection are thought of a little differently. So instead of doing the image gradient for edge detection, we use the idea of phase congruence, and I'll talk momentarily about it. Then, instead of doing non-maxima separation, we do something called gray level thinning, and then finally, we use hysteresis as in Canny edge detector to find the edges. So the idea of phase congruency is that perceptually salient features, so edges. When you look at something, the edges of what you see in your mind are very important features in delineating an object. So these edges are characterized by this thing called phase congruency, which is the degree to which the frequency components are all in the same phase. So this is a frequency domain analysis. So essentially what we do is we apply this notion of phase congruency, and I'm going to skip a lot of technical details here, but here is an image, here is the gradient magnitude of the image, and here is the phase congruency of the image. And as you can see, this is a much clearer delineation of the outlines. Okay, however, we're not done, you can see gaps here, and so forth, but now we can start seeing the individual parasites a lot more clearly. So after we do this phase congruency, so basically what we do is we apply every intensity value as a global threshold. We perform thinning on this image, and we superimpose the results. And that allows us to reconstruct especially those parts of the outline which are very weak edges, okay, which is where all the classical techniques have great difficulty. And as I said, we sort of reinterpret the Canny edge detector in each operation it is doing. So it does something called the non-maxima separation. So basically it preserves only the points which are regional maxima along the gradient orientation. So what we do instead is we do topological thinning of the image, and this is using only morphological connectivity information of the bases. So if you look at this, so this is what the Canny operator is doing, and as you can see here in this blown up image, you can see gaps between these pixels. So this is where the edge is missed. Okay? While over here, you will see that there are no gaps. So we get all these pixels. So these are very faint edges, which all edge detectors we have tried so far tend to miss. And I'm taking Canny as an example because it's a very well known and very well understood edge detector. But this one, the idea we are proposing, this has shown that so far we can find all these very thin edges. And the reason we are spending so much time, or I'm spending so much time talking about this, is remember again, we want to measure everything of the parasite. So for example, if I want to measure the length of the parasite, right, I need to measure the arc length of its boundary. And therefore it is very critical I get the boundary right, because if I don't, I'm going to have a mistaken measurement. And then when I start analyzing it, this is the algorithm is looking at it. The algorithm doesn't know that there is a mistake here. Right? So you can think, oh, this is a very strange, numeric value, and will make some conclusions which are going to be erroneous. Right? So even these little pixel level things are very, very important. And this is actually, if I step back from it and sort of talk to you as students in more pedagogical terms, one of the things you see in life is that the more refined the problem you try to solve, the little things matter a lot. It's not just one pixel here or there. That could be critical. Right? So the really good engineering solutions are the ones which not only do the big things very well, but also do the little things very well. So, I'm again going to skip a lot of things here, but here is sort of the flow of how this separation algorithm will work. So you take the data, you find the phase congruency, you do gray level skin thinning, you do hysteresis-based thresholding, you find the relevant edges, you do some morphological processing, and then you get your refined image by doing finally some thickening to make sure that you have not lost any pixels which were removed during some of these thinning operations. Okay. So this is what the final results, this is sort of representative of some of the final results we get. And it was actually incredibly good, because what we found out that earlier we were having large number of cases where these parasites would be sticking to each other and the classical edge detection techniques won't find them. But now we find that in the majority of the cases, and I'll give you some quantitative numbers later to give you a sense of things, we can separate the individual parasites out. The moment we can do this, we are in business. As a matter of fact, because I work in this sort of an area, I attend a lot of these biomedical imaging, bioimaging conferences and talk to a lot of people. And probably it's correct to say that 80% of the work presented, even today in all of these conferences, has to do with segmentation, separating individual things out. Right? So that tells you how tricky and how important this problem is. Okay? So once we can separate the individual parasites out, we are in real business. So next what we want to do is we want to track the parasites, which means that I want to establish correspondence between individual parasites in each frame of the video. Right? So as you know, a video is a collection of frames, right? And I want to make sure that I know that this guy in this first frame is exactly this guy in the 30th frame, and this guy in the 500th frame, and the 10 millionth frame, right? Because only then will I be able to coordinate the measurements I made of this parasite. Okay, so how do we do this? So this is actually an idea we've only slightly refined, but this was an algorithm which was developed by my good old friend Sonoma, and he was doing his PhD with me. And essentially the idea is that what we do is we use a graph theoretic technique. So the idea, what we do is the following. So we take the data from the segmentation level, then we construct what is called a blob level. So this is the architecture of our tracking method. So blob is nothing but a rectangular box around each segmented parasite. And then we use this graph theoretic approach, which was developed by Masood and Papanikolaopoulos, to establish correspondences between regions across frames. And then we assign identifications to each parasite, and then we sort of have a database. So we have an idea of the parasite, we have all the measurements of this parasite, and we are ready to do analysis. Right? So the ideas of this graph-based segmentation tracking approach is as follows. So what we do is we use a bipartite graph. So this is a graph where you have two sets of vertices and the edges are between these two sets of vertices. There are no edges inside that particular set, right? So we use an undirected bipartite graph to model the association between blobs and consecutive frames. So two blobs, which are connected by an edge, would be called neighbors. And basically this model is flexible. It allows splitting and merging of blobs. In other words, it allows for errors in our segmentation. Okay, if two parasites come very close together, then the blobs were merged from the previous frame, and the segmenter can't separate them out. And then this is one blob, it separates again into two parasites, and you have all of these sort of phenomena. Right? Very similar things happen, for instance, if you track pedestrians, if you go and track aircraft, and so on and so forth. So classical problems in image-based tracking in radar, and so on, and a lot of different areas. So here is, for instance, an example of one of these bipartite graphs. So you initially assign an edge between each pair of blobs that is sufficiently close to each other. And this is what is called the locality constraint of this algorithm. Then what you do is you systematically remove edges until the so-called parent constraint is satisfied, which means that a blob cannot simultaneously split an edge. And that would be an unnatural operation. Okay? So for instance, so you can see all these different ways you can split and remove these edges. And as you remove these edges, you compute a cost function. And the optimal value of this cost function tells you what is the best possible correspondence between these two frames. Okay. And the whole idea of this cost function basically is that you have minimum change in the area of the blob, and nothing moves very far. So it's a very simple idea, but it's a very rigorous way you can compute this, And Osama had shown that you can actually do this very, very efficiently. So you can do this at a frame rate. You can do this 30 frames a second. Okay. All right, so now we have tracking. And then, later I'll show you some videos and so forth. Now what we want to do is the really new part of this research, which is we want to convert now this information we have measured about each parasite after we have segmented and tracked it into what is called a time series. Okay? So what is the idea here? So think of this. Let's say that we have these two parasites. Okay, we are tracking them and we are measuring a bunch of properties. So for instance, we are measuring how the ratio of the straight line between its two ends and the curve, the geodesic curve which passes through the parasite between the two ends is changing. You're measuring what is the image difference, the number of pixels. You're measuring what is the mean intensity of the grayscale which is changing in these parasites, right? So as we track this, what do we get? We get, as this sort of video was showing you, we get this sort of a measurement which is going across in time. Okay? And this is what is called a time series. It is basically a measurement parameterized by time. Okay, now once we have this time series, and time series analysis in the last 10 years has become a very powerful tool in computer science. So, and it's done a little differently than how it is done in signal processing, and we'll talk about it briefly today. But now we have this time series, we have a very well represented form of all of this information. So we can analyze this time series and we can start finding very startling things about these parasites. So let's see, I have about 15 minutes. So what I'm going to do is I'm going to briefly talk about the key steps of time series analysis, just to give you an idea, and then I'll show you some results. So one of the problems with time series is that they can be very, very long. So let's say that you are measuring 30 times a second some particular value, and you're tracking this parasite over let's say 3 weeks. Right? It's a huge set of numbers just on one parameter, just one type of measurement. So what the time series community does, and they found this in other domains, they've not done this on parasites, but for example, they've seen this in stock market prices, or a lot of other things which have time dependency. So what they do is they represent the time series using symbols, the so-called symbolic representation of time series. So the idea is that you normalize the time series, then you sort of approximate it, using sort of a piecewise linear function, and then you assign symbols to each of these piecewise linear approximations under certain probabilistic assumptions. Okay. So this is a technique called SACS, which was developed by Imon Kyo when he was doing his PhD at UC Irvine, and Imon is now at UC Riverside. So it's very, very well, amongst the different techniques which are there for symbolic representation of time series, this has been quite well established, and it's got very good algorithms and very efficient algorithms to do this sort of analysis. So here is an example. You have two time series, and you represent them by these piecewise constant approximations. You convert them into symbolic representations, and then you can compare the symbolic representations using a lookup table, which represents the probabilistic distributions which were assumed when the symbols were assigned, and then you can have some sort of a distance function which tells you how similar or how different these time series are. And the reason that is of interest to us is that if we have two parasites which have certain behaviors which are represented by very similar time series, then we know that these parasites are acting very similarly, right? Without having to look at it. So we can do this all programmatically. So the problem in the SACS algorithm is that you have to manually tell it in how many pieces you want to divide this time series. Right? And basically, this is no way you can do it for the real world time series. It can't be done. So what we did, because we had real world time series, so we did the following. So what we did was we fitted straight lines to this time series by solving this optimization problem. So what you have is up here in the numerator, you have the L1 error of fitting a line segment to a set of points, and in the denominator, you have the length of the line segment. So basically, the idea is that you fit a line segment to the set of points as long as the error is small. Okay? And once the error exceeds the previous error, you break and you start fitting a new line segment. Okay? And so you now don't have to worry about how to figure out how many segments you need for this SACS approximation. And here are some examples where you can apply this and you can get your symbolic time series. And also, as I said, the dimensionality, the size of the data is now significantly reduced, which is property of these symbolic representations in the time series. All right, so now we have segmented the parasite, tracked the parasite, made all the measurements, converted it into this thing called time series, which the computer scientists seem to like a lot. Now let's start doing something with it, right? So let's see what we can do to, what sort of analysis of the data we can do. So one of the first things computer scientists love to do is they like to cluster stuff, because it tells them what is similar, what is different, right? So what we're going to do is we're going to cluster time series. Okay, and cluster simply means grouping things by similarity, right? So what are the things you need to do to cluster time series? Well, first of all, you need to define what is similarity. Okay? And mathematically, or even in signal processing or engineering, this is defined through a distance. So there are a lot of different ways you can define distances. You can have the Euclidean distance, which is a classical distance. You can have this min dist, which is this table-based distance, which comes from the time series analysis community. You can have something called the Levenshtein distance. Does anyone know what is the Levenshtein distance, by the way? Has anyone heard of this, the Levenshtein distance? It is related to something you guys in electrical engineering do all the time, or at least in coding you do all the time. How many of you guys have heard of the Hamming distance? Good, someone has. The Levenshtein distance is a generalized form of the Hamming distance. If the Hamming distance only applies to strings of same size, the Levenshtein distance can be applied to strings of different size. Then you have this idea of dynamic time warping, which is basically a dynamic programming optimization approach to find out the similarity between two sequences. Okay, so we have distance functions, and we can try all of these and figure out which one is the best. We have different ways we can do clustering, the actual clustering algorithm. Okay, so far we have tried two techniques. This is a classical hierarchical clustering, where you take similar things and slowly build up a hierarchy, like a tree, and I'll show you some examples. And we have more advanced techniques like density-based clustering, which again I'll show you some results. You can validate these clusters, but not go into this. There's too much of technical algorithmic stuff we don't care. So here's okay, this is basically more mathematical representation. This is the Euclidean distance measure. This is the min distance, which comes with the SAS algorithm. And I'm showing all this because I just want to show you how these different distance measures work in clustering this, right? So not everything works. Okay, this is the Levenshtein distance, which is basically a generalized form of what is called the edit distance. How many changes you have to make into a string to convert it into another string. And the two strings can be of different lengths. In the Hamming distance, they are the same length. Okay? And this is finally the dynamic time warping idea, which is basically you have some cost function, which is convex, and you optimize it using this sort of a tabular computation to find the optimal transformation of one string into another. Okay, so these are the different types of distances. If you're not haven't come across them, take an algorithms class. They will not teach you all of this stuff, and you'll be better off. And then it gives you that. So here's an example. Okay, so here are the time series of a controlled set of parasites, normal parasites, no drugs. That's controlled. Here is the time series of parasites which were exposed to the drug Levenshtein. Okay, so this is 1, 2, 3, 4. So these numbers tell us which parasites were controlled. These are the Levenshtein parasites. These are the parasites which were exposed to the drug Praziquantel. Okay. Now, a proof of concept would be to see if our choice of how we represented and what distance we use get this groupings correct, right? So here are some examples. Here is the Euclidean distance. Here is the min distance, which comes with SAS. Here is the Levenshtein distance, and here is dynamic time warping. And if you see actually of these, the way to read these dendrograms is you see here. So your 1, 2, 3, and 4 are clustered together. Okay, so this is the control. Okay, then 10, 12, 11, and 9 are clustered together, which is over here, and then these guys are clustered together, 5, 6, 7, and 8. Right? So this is actually the distance in this example, but also in many, many other examples, which turns out to be the most accurate. So these other distances for this sort of data don't work out very well. So this is, I point this out, especially in an engineering department, in a math department, I could store for seconds, but this is trial and error of engineering. You have to try a lot of different things. They're all valid ways of thinking about it, but for given the nature of the data, some of these techniques will work better. So there's a lot of, sort of seat of the pants hard work involved in, tuning these systems. So, here is, the clustering algorithm. So this is this idea of this agglomerative clustering. You build this tree. Here's the idea of DB scan, which is basically find the dense spots in the distribution and you cluster them together. We'll not worry about the details, doesn't matter. Now, what I want to show you, so this one last thing, which is that once you form these clusters, you can define a representative time series of the cluster. You can say how what would be the most common way all the parasites in a particular cluster be. Okay? And then, I'm not going to go into this detail, but I'll show you some examples of these representative parasites from these clusters. And then you can see why this is an interesting way of thinking about it. So some example results. So the segmentation, how does it do compared to many other techniques? So here are some numbers in terms of average runtime. So the average runtime of this method is actually the fastest of all the ones we have tested. So these are all published methods. This actually, JSA, comes from a very renowned electrical engineer, a very good friend of mine, Manjunath from UC Santa Barbara. This is a very, very good algorithm, but did not work very well on our data set. In terms of precision and recall, the algorithm I showed you, this is yet unpublished. This one was published in ISVC. So we are getting precisions of about 90.27% and recall of 91%. So 90 plus, okay. Very, very good. We can live with those numbers. Here's actually another way of showing you that this new segmentation algorithm I briefly talked about, why this is good. So this is this old algorithm we had, which was published in ISVC, and this shows the segmentation of 2 consecutive frames using this old algorithm and the overlay of these segmentations. And you see a lot of stuff around, right, which shows that just in 2 consecutive frames, the segmentations are somewhat different. So when you overlay them, then you get all this extra stuff, right? So in 2 very consecutive frames, the parasites will move a lot. Right? So the idea is that if you segment with high stability, you should get basically the entire parasite, which is what you see. So it's not just the numbers which are good, it's also a very stable algorithm. So small changes do not bring large changes in the segmented results. So here's a tracker. Okay. So basically this is a bunch of videos based on these different drugs. So here's a segmentation and this is a tracking. So in the first case, 73% of the parasites were tracked properly, which is a bit of a lower number. In the second case, 88%, 84%, 87%, 81%. So 80, to mid-80s or high 80s is what our tracking accuracy is right now. Okay. Which incidentally is significantly better than what humans do. So we're already better than humans. So here is a very, very interesting thing. Okay? And I'm going to basically end with these slides here. So this is what we did. We found the time series, we clustered the time series, and using that algorithm, which I didn't tell too much about, but I'm happy to explain to you offline if someone's interested, we found representatives of the clusters. Okay? So we found out, for instance, the control, the parasites which were not exposed to any drug. Okay? As you can see, there are different clusters in the control. So these are representatives of each cluster. And you can see these guys look very different. Right? So the point here is, and this is something some of my collaborators have been working on the disease for the last 30 years. And they never, they used to see this, but they never had a clear idea of how systematic this thing was. So there's a lot of variability of these guys. So here is the parasite being exposed to this drug Praziquantel. Okay, so this is after 7 days, and you see 3 very different clusters of parasites, and these are representatives of each of the clusters. And you see very, very different looking parasites. Okay, so the drug is acting very differently on different parasites. Okay. What this means biologically, what can we do with this in terms of drug discovery, I do not know yet. Okay, the result is so new and it's so surprising to some of the parasitologists. We haven't even figured out what to do with it. Okay? But the point I'm trying to make here is that by using engineering and computer science, we are getting perspectives on a disease which some people have studied for many, many years, which are very unique. Right? So us coming, as us engineers coming into play here, is providing the type of knowledge that the biologists, the parasitologists, the drug discovery people just did not have earlier. So, I promised Ali that I will finish on time, and I'm going to hold on to my promise. So what have I talked about here, right? This is not really, forget this conclusion, it's not about what we have done and why this is so interesting. What have I talked about for you guys, for students here? Okay, so first of all, in terms of the techniques, what I've hopefully tried to show you is an engineering solution where we have taken this highly complex problem of phenotyping a parasite, which has many different behavioral patterns when it's exposed to a drug, and causes a very important disease, in which normally would take trained PhD human scientists sitting in a microscope for hours and hours and days, okay? And we have tried to make it completely automated. And the method, now that it's been implemented, has started showing startling things. And the fact that there is variability in the parasite population, which is not at all surprising, but now we can quantify this variability. We can not only find it, we can statistically quantify it. I've not shown you some of these measures of cluster quality, so we can actually ascribe very rigorous statistics to this. And what this would allow us to hopefully do is to screen new drugs and make this sort of data available to the entire community. There is no phenotypic database available to parasitologists in the world at this time. So hopefully in the next 3 or 4 months, we're going to bring one of these databases online where anyone can go and download all of this quantitative information about these parasites. But all of this is aside, doesn't matter. I mean, if as intelligent people try to work on a problem, we expect to have good solutions, or we hope to. But what is very important, especially for those of you who are students here, I hope you try to, you've seen that this is something very, very different. I did not talk completely about parasitology. I did not talk completely about chemistry and drug discovery. I talked a lot about engineering, about signal processing, image processing, some algorithms, okay? That's the bias of my particular field I'm coming from. But the point is, we are taking knowledge and ideas from wherever it matters to solve the problem. That we're not constrained in the straitjackets of classical disciplines. And it is my firm belief, and it's my firm experience, having worked for so many years in the industry, that when you guys go out and become the specialists of tomorrow, you should try to become like this. Try to solve problems. Don't worry where your ideas are coming from. It's irrelevant. As engineers, we are here to solve problems. That is our contribution to society. And hopefully this was a small example. So with that, I'll conclude and take your questions.